At the University of Bristol, a new life sciences building is currently being constructed and is due to finish by the end of 2013. As well as having the new building, a piece of artwork will hopefully be installed. This piece will be a collaboration between artists and scientists, demonstrating the diversity of the work going on in the biology department and the creativity and skills that involve combining this with art. The artist we are working with is Thomas Saraceno, an Argentinian who has previously been involved in creating incredible structures, combining live plants and man-made works. It is hoped that the piece will be installed into the main atrium of the Life Sciences building, and as you can see from the concept designs, it will look quite spectacular once completed. The structure will be formed from cables in a spiderweb-like net, linking 5 glass spheres together, as we aim to link the different areas of biology. Dr. Heather Whitney has been working with Shirin and I to experiment with some initial ideas as to what may go into the spheres, whether biological or biomimicry. Much experimentation will be needed. So, idea number 1. We thought there was much potential for growing algae in these spheres, as they would require limited maintenance, have quite spectacular colours of greens, reds and browns, and also we like the idea that as time passed the algae would grow so the art would be changing. Our first thoughts were to get some brightly coloured algae, some agar gel, and then swirl the gel up the sides of a spherical flask so that the algae would slowly grow up the sides following the pattern of the agar. Idea number 2. This involved growing some mushrooms and potentially using agar instead of soil, as it may be more aesthetically pleasing and probably a lot lighter. The variety of colours and morphologies of the different types of fungi really made this idea potentially very exciting. Idea number 3. Next, we thought of using moss balls, as they look very attractive and even look better with a bit of movement in the water. These moss balls are used in aquariums and fish tanks and actually outcompete other types of algae, so keep the water looking clear. As we were unsure as to whether there would be an electronic source to the sphere, we thought that having a solar-powered motor would be a possible solution. Idea number 4. This idea came from a thought about the fact that we regularly see plants growing from pots on the ground. However, we don't often see them above us, let alone growing upside down. So, as we knew Stuart Casson was growing Selaginella, which had survived for a year on agar in a sealed container, we thought this would be a perfect plant to use in our project. Idea number 5. One of the problems that needed considering was the issue of maintaining whatever we had put into the spheres for the duration of the piece. So, we looked into some potential ideas that didn't involve maintenance. One thought was to use the fact that with microscopic organisms, you can't see them normally with the naked eye, so you would need to do a model to show what they look like anyway. So, looking at microscopic organisms, we picked a few interestingly geometric ones, like the radiolarian. The other part of the idea was to incorporate some of Heather's work on iridescence and look into different organisms with iridescent shells or such like. Idea number 6. After having looked at some of the climbing plants that are present in nature, we thought it would be a good idea to have an open-top sphere. Out of the sphere would grow these climbing plants, and as they grow, their tendrils will wrap around the spiderweb net structure, blurring the lines between the living plants and the man-made web. Idea number 7. A Vinogradsky column is a perfect example of a changing biological environment in a contained area. Soil and water samples are taken from local spots which contain different volumes of microorganisms, other biological material, and gases. As time goes by, the different layers separate out depending on the relative densities, and the microorganisms grow, producing different colours. If we use this, it would be another changing sphere that developed over time. Idea number 8. This idea would involve getting ants and allowing them to create their own art. This links in with the idea that in nature we see many structures that are created by organisms, which are huge feats of engineering and science, and yet they do it just as a part of their everyday lives. Hopefully the series of interconnecting tunnels that they will create would look quite interesting. So, that was an overview of a few of the main ideas we have had for the project. The next film will include our expeditions outside of the biology department to do some research out in the field, testing out some of our ideas, and then hopefully a conclusion on which ideas were most successful. Thanks for watching!